In the last few years, we started to think, should we really do bone marrow biopsies, and are we getting all the information from the bone marrow biopsy? And I think the revolution of liquid biopsies or blood biopsies is very important, and I think there are multiple parts in that. There is the cell-free DNA, but there is the circulating tumor cells, and then there are so many other things, proteomics, metabolomics, exosomes, microRNA. There is so much in the blood that can give us a picture of the whole system, of what's going on in the cancer as well as the immune system of our patient. And I think the future is going to get better and better in the technologies. We are already sure now that we can use circulating free DNA to mimic or to show us what's going on in the bone marrow, and potentially even show us certain clones that were not seen in the bone marrow biopsy, which is very exciting, but we need to improve on those technologies. And in the next few days, we will show some of those newer technologies of how we can actually have a deeper coverage to understand better which genomic alterations happen in the peripheral blood and how can we detect those in those patients.